Good evening, ladies and gentlemen. Welcome to BrisScience for December 2017-18. I am your host for tonight, Joel Gilmore, and BrisScience is the University of Queensland's free public lecture series on science, where we bring not just the best scientists, but also the best communicators to share their research with Brisbane. We're hosted here at The Edge, part of the State Library of Queensland, and they've been our wonderful venue partners for the last few years. So can we join in a round of applause for The Edge? We appreciate the work that you guys go into. I'd also like to respectfully acknowledge the traditional owners of the land on which we're meeting tonight and pay my respect to elders both past and present. So tonight we are having a very special BrisScience where we're talking about artificial intelligence, but we're also talking about homegrown intelligence and what we still have to learn from our very own brains and what that might mean for computer science and for artificial intelligence in the future. And we have not one but two fantastic speakers this evening who I'll introduce in a moment. A couple of little bits of housekeeping first. If you've got a mobile phone, if you could switch it to silent, and if you could load up Twitter, ready to ask questions using the hashtag BrisScience, and we will take those questions at the end. Or if you would rather go traditional pen and paper, we'll take questions through those question slips you received on the way in, or on the back of a $10 or $20 note. Write those questions down, and we will come and collect those questions at the end and ask as many of those as we can. Afterwards, we do have some food and drink, and there'll be a chance to have some more discussion yourself and to ask our scientists tonight some more questions if you do. I think that's all of our housekeeping for now. So I'd like to introduce our two speakers. First up, we have Professor Geoff Goodhill, who originally studied in the UK maths and physics, which I heartily approve, but then also got into artificial intelligence. And he became fascinated by understanding the algorithms that the brain uses to achieve its astonishing feats of computation. He now holds a joint position with the University of Queensland's Queensland Brain Institute and the School of Mathematics and Physics. So holds a diverse position there. And joining him tonight is Dr. Lilach Avitan, who is a computational neuroscientist, also from the Queensland Brain Institute. And she investigates how our brain processes information in order to understand brain function and how to build more intelligent computational devices. She was also selected recently by Science and Technology Australia to be a superstar of STEM as one of Australia's most dynamic female scientists. So Geoff is going to kick us off, and I'm going to hand over the evening to them because they're going to run it themselves, and there's some great toys up here. So could you please put your hands together tonight for Dr. Geoff Goodhill and Dr. Lilach Avitan.
Joel, thank you very much for that kind introduction, and thank you for coming. So this is a plastic model of a real human brain, and your brain consists of about 10 to the 11 neurons. That's 100 billion neurons. And your brain makes up everything that makes you you, all your thoughts, all your desires, all your plans for the future, all just contained in this about 1.5 kilo thing here, which we really don't understand much about. We do understand it's capable of remarkable feats of computation. And so, although there's been a lot of interest, a lot of excitement, a lot of progress in artificial intelligence recently, there is still an awful lot we don't understand about what makes your brain intelligent and what makes your brain more intelligent than any artificial intelligence device that's currently been developed. So we're going to talk more about artificial intelligence later. But first, I want to give you some examples to show you just how amazing your brain is. So I'm going to pick on somebody in the audience. How about the chap in the red T-shirt here? What's your name? Sorry? Calder. Okay, now Calder, I would like you to catch. Fantastic. Now, let's just think about all the things that went in Calder's brain there. So, firstly, he was able to decode this continuous stream of sounds that's coming out of my mouth. So, if you look at this on an oscilloscope, it's just a continuous waveform. I am not speaking like this. Through Calder's extensive knowledge of the English language, he was able to divide that up into words. He was then able to understand what all of those words meant and what meaning I was trying to convey by those words. Now, that's a very non-trivial task, and I'll show you another example of that later. So This illustrates a very important point, which is that it doesn't feel like hard work. You're probably thinking, come on, that's easy. I do it all the time, right? But it's not easy. The problem is your brain is optimized to solve exactly those kinds of problems. And that makes it very difficult to understand quite how difficult it is to program a computer to solve that kind of problem. So anyway, so come back to Calder and his remarkable feats of computation just then. So he understood the word, so that happened in the part of his brain, the auditory cortex that processes sounds. And then some signals went to the part of the brain which is involved in decision making, and Calder decided that what I wanted him to do was position his arms to catch this object. So he then sent commands to his muscles to move his hands into precisely the right location to catch that object. Now, this involved an awful lot of prediction about the trajectory of that object. So I threw it straight towards him, right? So he didn't see a nice trajectory going across the visual field like that. He just saw this object coming towards him, getting larger. Now, that's very little information to predict where that object was going to be. But he did it perfectly. He sent the commands to his muscles, and he moved his hands. Now, moving your arm to a particular location is what's known mathematically as an ill-posed problem. What that means is there are infinitely many solutions. So I can get my hand from here to here. I can do that, or I can do that, or I can do that, and so on. So we don't know how you choose what trajectory to move your hands. We also don't really understand how you can coordinate all your muscles to, once you've chosen a trajectory, to move your hand in precisely that trajectory. Anyway, Calder was able to do it apparently effortlessly. And then while that object was in flight, he made a number of inferences about the properties of this object. So firstly, he estimated the weight of the object. So if he had reason to think I was throwing a gold brain at him, no such luck, I'm afraid, he would have positioned his hands to catch a very, very heavy object, and that would have been different from the way he positioned his hands just then. Similarly, if I just pulled this brain out of a vat of olive oil and thrown it at him, he would have positioned his hands to catch a very slippery object, and that would have, again, have been different. So all that computation happened in Calder's brain in the space of about half a second, and the power consumption of your brain, whether you're catching an object or not, is about 20 watts. So you think of the power consumption in this room at the moment, it's probably thousands of watts. So Calder was able to do all of that using a power of 20 watts. I mean, that's an awful lot less than these computers here. And did it feel like hard work? No, it felt easy, right? Because that's the kind of thing your brain has evolved to do, the kind of computations it performs very, very easily without apparent effort. Now, Calder is probably also an expert chess player. He probably plays a musical instrument as well. He can do many, many different things. And for some of those, like chess, dedicated artificial intelligence programs are now better than any human at those things. But if you take the entire package of everything Calder can do, we are nowhere close to that with any artificial intelligence device. So nowhere close to this kind of general intelligence which can solve all those different kinds of problems and interact with the world as effortlessly as Calder just did. So could you just chuck the brain back to me? Thank you very much. And let's have a big round of applause for Calder. Thank you. Okay, so what are these signals that are flying around in your brain that were flying around in Calder's brain just now? Well, the brain transmits information between the neurons by a digital code. So they're basically spikes of electrical activity, and it's both the rate at which those spikes occur and the timing of the spikes which seems to transmit information in the nervous system. So there are billions of spikes flying around in all of your brains right now. Even just sitting there at rest, even if I wasn't talking, there would still be lots and lots of activity in your brain as you process all the information around you in the world. And some of the research we're doing in the lab is trying to understand the nature of that neural code, how neurons convey information through patterns of ones and zeros. And that's what my colleague, Dr. Avitan, is going to talk about shortly. But first, I wanted to directly demonstrate those pulses of electrical activity to you. And for this, I need a volunteer from the audience who's willing to come up on stage here and be... Oh, did I see a hand from the young man in the red T-shirt there? Please, come on stage. Now, what's your name?
Neo, N-E-O.
N-E-R, okay?
Oh.
Oh, N-E-O, sorry, Neo.
Yes.
Fantastic. Okay, now come and stand here. And what we're going to do is we're going to hook you up to this device. And let's see, we're going to put some electrodes on your arm. And what we're going to do is we're going to record, give me this arm, yeah. We're going to record pulses of electrical activity traveling down your arm to control the muscles in your hand and your fingers here. And so normally it's quite difficult, obviously, to record from individual neurons. You can do that from people who are having brain surgery because they need to have surgery for epilepsy, for instance. But you can get some sense of what's going on, even through the skin. So this is what we're doing here. So we're going to wire you up here and here. Now, I would hope that you have good insurance, but I'm sure it will be fine. Okay, so what we're going to do is I'm going to open up an app on my phone, if it's still working. No, of course, the app has died. Oh, no, okay, so there we go. And we're going to plug this in here. And and we're going to plug this in here. I'm afraid my talks are an AV guy's nightmare, but thanks, Todd, you're doing a great job. And so we're going to plug this in here. And we're going to turn this on. Okay. Now then, so that's just background activity. Now what I would like you to do is squeeze your hand. And you can see right there. So what you're seeing is the echo of electrical activity traveling down Neo's ulnar nerve, telling his hand to squeeze. So just relax. And the activity goes back to normal. Squeeze again. Get more activity. Relax. So we're actually directly measuring the commands that Neo's brain is sending to his hand there. So let's zoom in a bit. And let's threshold. So what we're going to do now is squeeze really hard. There we go. And see, what we're doing is we're averaging over lots of those pulses. And you see, the one you see in the middle there is the characteristic shape of one of those pulses. So if you can see the width there, it's only about 1 or 2 milliseconds wide. So each of those pulses is very, very short, about 1,000th of a second. And yet, each of those, together those pulses are giving very precise instructions to Neo's hand. So just relax again. We'll just do one more thing, which is, let's go back to recording again. Now, so we saw that if you squeezed your hand, then we get lots of activity. Let me just zoom out again. Now, what do you think would happen, so just relax, what do you think would happen if I squeezed your hand instead?
Well, nothing would happen because I'm not doing it myself.
Right, so let's see. Let me try that. So you just stay completely relaxed, and I'm going to squeeze your hand instead. And what's happening?
Nothing.
Just as you predicted, absolutely nothing. So well done. So the reason is because... Yep. The reason is because there are no signals traveling down your arm because you're not telling your hand to be squeezed. I'm doing the squeezing. Fantastic. Very good. Thank you very much, Neo. So we'll just unhook you here, or just take this off and just unhook you. Thank you. So let's have a big round of applause for Neo. Okay, so hopefully we can get this going again. Great. Okay, now, this is the bit I really love, which is that the shape of those the individual spikes, they're sometimes called action potentials, is to do with the flow of ions across the neural membrane. There's a whole bunch of complicated biology. But in the 1950s, 2 neuroscientists called Alan Hodgkin and Andrew Huxley were able to write down a mathematical model for the shape of those spikes. And these are the so-called Hodgkin-Huxley equations. So let's look at what they predict for the shape of these spikes. So one of those pictures is the theoretical prediction, and the other picture is the experimental the experimentally measured shape, and I can never remember which way around they are because essentially they're almost identical. So those equations almost perfectly describe the shape of that nerve impulse. And this shows how we can, despite biology in general being very messy, there's some parts of neuroscience where we can develop a very precise mathematical understanding. And an important point I'm going to make later on is that through that precise mathematical understanding, that is really useful for building artificial intelligence. Because to build artificial intelligence, you need to program a computer to do something. And so you need to turn biology into mathematical type information for the computer to be able to do something with it. So that's very important for a number of things. So it's important, for instance, for building brain-computer interfaces. So reading that code, for instance, using those kind of electrodes or electrodes actually implanted in the brain, reading the code and trying to use that to drive, for instance, prosthetic arms if you're paralyzed. And it's also very important for developing artificial intelligence, which I'll say more about later. So I want to introduce three important areas of research here. So first is computational neuroscience. So that's what I do, that's what Lilach Avitan does. And that's about trying to understand the computational principles that biological brains use. So that's the Hodgkin-Huxley equations are an example of that, trying to understand what kind of mathematical computational principles are involved. Artificial intelligence is about building intelligent machines. And then another area, which you might have heard of, is neuromorphic computing. That's how you build hardware which was optimized for biological computation. So once we understand more about biological computation, you can simulate anything on a conventional digital computer, but it's much more efficient for some kinds of computations than others. And the kind of computations that are going on in your brain are, take a lot of energy on conventional hardware. So this laptop is using hundreds of watts. My brain is using about 20 watts. And this has a vastly large number, greater number of processors than this laptop. And as we saw with Calder's brain, is capable of many more interesting things than this laptop is. And yet, nature has found a solution which allows all that computation to occur using an incredibly small amount of energy. And so if we can unlock what the secrets of that energy-efficient computation is, it will obviously be very important for building better computers. Okay, so let's just review now some of the things that make the problems that your brain solves very, very difficult. As I say, it's hard to appreciate because your brain does it so easily. So one of them is understanding visual images. So one of the problems with understanding visual images is that it's a three-dimensional world out there. You've got a two-dimensional image on your retina. Now, it turns out that for every two-dimensional image on your retina, there's actually an infinite number of three-dimensional scenes out there which could have given rise to that image. And so every moment you're looking at the world, your brain is solving, again, a type of ill-posed problem, a problem for which there are infinitely many solutions, and yet it picks, in some sense, the best one we can all agree on. And so I want to show you an example of that. So there's a very nice competition that runs each year called the Illusion of the Year contest. And this is an example of one of the winning entries this year. So there shouldn't be any sound on this. So what we're looking at here is, so these are, oops. There we go. These are three objects, and they all look the same, right? Pretty much. And what we're going to see is we're going to reveal some mirrors back here. So this is made by some guys in Japan. And so in the mirror, it looks the same except the rooster's disappeared. In here, it looks completely different. In here, it looks completely different. So what is going on there? Well, in a moment, they're going to rotate this table, and you will see that, in fact, these three objects, which all looked exactly the same from one perspective, actually have three completely different three-dimensional configurations in the world. And so this is an example. So this is obviously a very contrived setup, but it's exactly the same issue that you're solving all the time, in that when you looked at those, you didn't see the thing that's really there. You saw the thing that seemed more likely to be there based on all the experience your brain has in the world. And so you're constantly solving these ill-posed problems. I'll just give you an example of language as well. So that's understanding vision. We talked earlier about understanding language. So consider a very simple sentence like this. So for a computer or for you to understand this sentence, you have to understand what this word they refers back to. Now, it refers back to something previously in this story here. So most of us would agree that in this context, the they refers to the politicians, right? But we can change one word at the end of the sentence, and now most people would agree that they now refers to the crowd, right? So how do you make those decisions? Well, it's based on an enormous amount of sort of deep knowledge of how the world works, right? I mean, imagine trying to program into a computer rules that would tell it, well, in this case, the they means the politicians. In this case, the they means, so in this case, the they means the crowd. That's a very subtle kind of knowledge in the world. And in the early days of artificial intelligence, people thought they could build intelligent machines by just writing down every single piece of knowledge that humans have about the world. And although that effort continued for a while, people eventually gave up because they found that's just not the way to do it. You just can't write down enough information that humans have acquired just through a normal, just growing up in the normal world, all the incredibly deep knowledge humans have acquired about the world. So what is perception then? How are we solving these problems? So a very powerful idea in computational neuroscience is that perception is actually statistical inference. And a key idea was actually introduced by the Reverend Thomas Bayes about 200 years ago. So he was an English clergyman. He was also a gambling man and became interested in the laws of probability. And he proved a theorem now called Bayes' theorem. And this is critical because it tells you how to update your beliefs about the world or integrate new data with your prior beliefs. And this is his equation here. I won't go through the details, but essentially this is your model of the world, this is the data, and it says, given some data, here's how I should update the probability that my current understanding, or compare the probabilities of different models of the world. Now I want to illustrate to you that we can use this to solve a lot of different problems in neuroscience. And one of the problems we've been interested in neuroscience is understanding how nerve fibers are guided to their targets in the developing brain. So neurons migrate, they have to find appropriate locations to grow to. So this is a growing nerve fiber. This is 1,000th of a millimeter across, and the tip is about 100th of a millimeter. And although you can't see it, coming out here is a gradient of what's called a chemotropic factor, which is attracting the tip of that nerve fiber up a concentration gradient here. So how is this tiny device able to detect a concentration gradient, that is a change in concentration across its spatial extent? Well, we appealed to some computational ideas. So here's a simple model of what the tip of this nerve fiber looks like. We have some molecules from the gradient which are binding to some receptors here, and they're going into this complicated signaling network. So we suggested that the tip of the nerve fiber might be appealing to the good reverend and performing Bayesian computations. And from that, we were able to write down a formula which predicts how accurately the tip of this nerve fiber should be able to detect whether which way the gradient is pointing. And we compared that with the experimental measurements, and we found that gave a very good prediction. So we can use these ideas of statistical inference, not just in understanding how you catch a ball, but how nerve fibers find their targets during neural development. So to lead into Lilach Avitan's part of the talk, More recently in the lab, we've started looking at using Zebrafish as a model system. So Zebrafish are a beautiful system to use in neuroscience. They grow up very quickly. This is a 5-day-old zebrafish. It's just a few millimeters long, and what it's doing here is hunting a paramecium. So 5 days after being a single cell, the zebrafish has already grown up to an animal that can catch a fast-moving prey. And one of the things that makes zebrafish very exciting to study is that young zebrafish are essentially transparent. So this is the head of a young zebrafish. These are the eyes here. The red is just a structural marker, but the green is labeling individual nerve fibers growing from the retina into this part of the brain called the optic tectum that processes visual images in zebrafish. And because it's transparent, we can embed the fish in agarose under a microscope and make long time-lapse movies of these nerve fibers growing. So this is a movie of an individual nerve fiber growing in the zebrafish brain. It's growing over the surface of the optic tectum here, and this is over several hours of development, and it's trying to find where it should make connections to. And we modeled this behavior mathematically using the kind of tools I've just shown you. Now, more recently, what we've been doing is embedding fish in agarose again, but using transgenic zebrafish, where they have a fluorescent label in the neurons, which allows us to see these pulses of neural activity. So when there's a pulse of neural activity, calcium flows into the cell, and so we have a fluorescent calcium indicator, which tells us which neurons are active. So here is a movie of a good part of the brain of a zebrafish, this part here, and each little green blob here is an individual neuron. So we've moved a bit beyond recording from Neo's arm here. We've got a fish under a microscope where we can actually record at single cell resolution the simultaneous activity of many, many neurons in the zebrafish brain. And so this is a very exciting opportunity for us to try and understand the principles of neural coding in this organism. And we believe that these principles evolved a very long time ago and are conserved over fish and humans. So at this point, I'm going to hand over to my colleague, Dr. Avitan. So I'll just unplug him.
All right, so as Geoff showed you, our brain speaks with these spikes, right? And this is the neural code. And everything that we sense, we feel, we think, we plan, everything is coded with these tiny spikes. How is the brain really holding this information? How do neurons speak to each other? How does the brain drive behavior? That's one of the biggest challenges in neuroscience. And today I'm going to show you how we are tackling these questions in the lab. So, not long ago, we believed that everything, all the information, resides within the single neurons. So here you see an action potential, a spike train. So these are the spikes that we recorded not long ago from Neo's end. But now, without the noise. So whenever there is noise, there's just blank, and whenever there is a vertical bar, that's a spike. Okay, and we believe that this is where the information resides. And in fact, indeed, a very famous study published in Nature showed a single neuron from a human brain that was very, very particular in its response. And this neuron really preferred Jennifer Aniston's face. All right? So here is the spike trains. These blue bars are the spike trains. Okay, and you can see that it didn't really matter whether what her hairstyle is. It didn't really matter what the lighting conditions is, or even the angle of view. This neuron really preferred Jennifer Aniston. And in fact, when we showed this neuron, or this human, photos of Jennifer Aniston with her partner, this neuron didn't really like it much. In fact, even more than that, this neuron, when we try to stimulate it with very familiar faces from different disciplines, complete silence. All right? Also not for different animals or different architectural structures and so on. So this neuron was the Jennifer Aniston neuron. And this has led us to this notion of a grandmother cells. Okay? These are cells that will be active when we see our grandma. However, this notion has a little bit or many problems in terms of design and in terms of efficacy. Well, imagine what would have happened had this neuron was damaged. What will you not be able to recognize Jennifer Aniston anymore? And this has led to many questions. And one of the problems in this single neuron coding is that these neurons are very unreliable. They vary in their responses. So if I show a single stimulus and record the activity from the brain, you would get one pattern. But if you show over and over the same stimulus, you will get actually will never get the same pattern. All right? So this led us to believe that actually the information must reside at the population level and not at the single cell level. So this has a bit shifted or diverted the attention of neuroscience instead of recording single neurons to record from populations of neurons. So over the year, technology has allowed us to record from increasing number of neurons. Back at the 60s, we were recording from a single neuron, maybe pairs of neurons, and today we are recording at about hundreds, maybe 1,000 neurons simultaneously. And this exponential growth shows that we are doubling the number of neurons recorded every 7 years. And this led us to think that if we wanted to record from the whole brain, the whole human brain, at the single cell level, it will take us about 200 years. Not a problem. The thing is that even if we had these data sets recording from the entire brain at the single cell level, at the moment, with the current computational capabilities we have, we really cannot handle this kind of data. Okay? And this is where animal models come in hand. And so today I'm going to show you some work in the lab we are doing, where we are standing here at the very right end of this graph. So we are using the zebrafish, as Geoff mentioned. This is the adult zebrafish. We are using it as a larvae. This larvae is, all of this brain is labeled with green marker, and this is its brain. Now, what we do in the lab, we found, first of all, that even when in the absence of any visual cues or in the absence of any cues, our brain is remarkably active. And the question is, what is the brain doing? We believe that it has to do with the computation. And so we started looking at ongoing spontaneous activity in the fish. So the way we do it in the lab, we're having a fish in a dish, and the fish is sitting under a blob of gel. The gel is set, and the fish cannot move. And then we cover it with water. And while the fish is sitting in the dish, thinking fishy thoughts, we are recording the activity from the brain. Okay? And this is the visual brain of the fish. And this is how a brain with no stimulus looks like. So every precious pearl here is a neuron. And you can see from time to time single neurons are active. And at certain time points you can see groups of neurons that are active together. And the real question is, what is going on there? What is driving this activity? What does it mean? What is the function of this activity? And so we really asked, how is this activity changing over development? And what affects this activity? And so we first looked at very simple measures to see what happens to the amount of activity over development. And what we saw was we looked at the fish at different ages, from 4 days post-fertilization until 9 days post-fertilization. And what we saw an increase in activity from day 4 to 5, and then it remained up high and then declined at day 8. But recording from hundreds of neurons simultaneously, a lot can be discovered. So what we did is we looked at the relation between the neurons, looked for correlations between the neurons. Neurons that tend to fire together, they could be also somehow connected. And so we described the brain as a mathematical structure called a graph. Okay, so here every neuron now is a black triangle, and every edge here now is between two neurons represent high correlation, some relation between the neurons. And we call that functional connectivity. What we found was that this functional connectivity increases also at day 5 and then decreases again. So again, something is happening over development. Now, if we look at this graph, and we saw that also in the movie, that these neurons, they tend to fire together, the clusters of neurons, or we call them neural assemblies. And when they fire together, using these very recent techniques from graph theory, now we have a lot of a really great renaissance of techniques for graph theory, simply because there's a great interest in mining data from social networks. So we can think of this graph as a social network and find those interconnected groups of neurons that are firing together. And when we applied this technique, we could identify, first of all, how many assemblies we have, and even this number peaked at day 5 and then declined. So there is something happening over development. The question is, is this some genetic program that the animal is going through, or is it something to do with the environment we live in, or the environment the fish live in? And so what we did is we altered the visual input of these fish while they are in the lab. So we had a group, we had 2 manipulations. We had the normal reared fish, and they were sensing day and night, but we also took a set of fish and we reared them in the dark. From the day they are born until day 6, they were in the dark, no visual cues. And then we also wanted to have a fish that sit in a complete light, but the problem is that I wanted to avoid any contours or any features in the visual cues. So I came with this solution of putting the fish in these bulbs, so they can see no contours, no features, so they are living in a featureless environment. And so we looked what happened to the functional connectivity after these 2 manipulations. And what we saw was that after dark rearing, this functional connectivity decreased, so the brain was less wired. Okay? Something was missing in terms of wiring, meaning that there was something else is driving this functional connectivity. And when we looked at the functional connectivity of the featureless reared fish, we saw an increase in functional connectivity, which means that this fish did not undergo the refinement needed to become similar to the normal fish. And so we said, okay, so there is some changes happening in our brain, and these are happening all over development. And we know now that the visual environment is important, but does this affect behavior? And so we had to look at the behavior of the fish. Now we chose one of the very prominent behaviors in the fish, which is the hunting behavior. So the fish are born with the yolk sac, and they are feeding through the yolk sac until day 5. But from day 5, they have to start and hunt for their food. And I'll show you how a 4 millimeters long animal is approaching 120 microns single-cell creature that is moving in the dish and feeding. Now, since the animal is transparent, you can actually see the food running through the body of the fish. All right, so we have a behavior. Now we wanted to check whether our manipulations affect the behavior. So we took the fish that were reared in the dark and the fish that were reared in normal conditions. We placed them in a dish with 50 paramecia, and we let them feed for an hour. And after an hour, we checked how many paramecia there are in the dish. And this gave us an indication how well the animal did in feeding after this visual manipulation early in development. It's very fun to look at these movies. So what we found was that whereas the normal reared fish ate roughly half of the dish, the dark reared fish didn't eat a lot. So it's not only that the brain is affected by our visual experience, it's also the behavior is affected by visual experience. And even when we let these two sets of fish to recover in normal conditions, still the dark-reared fish were performing less well than the normal reared fish. And this adds to a great body of work saying that, also in the mammalian brain, saying that there is a very critical time window over development, that our brain is very sensitive. And if it doesn't get the right stimuli, it will not wire up correctly. And even if the stimuli is then introduced, it will never be wired up correctly. So this is... and this could be a critical period as well. And the fish can be a good animal model to test that, even in greater detail than the mammalian brain. And if you want to learn more about this work, this is recently published in the Australasian Science in the latest issue. Now, obviously, this is where the fish is sitting in this dish and thinking his fishy thoughts. But really, all of us are interacting with our world, right? So how can we let the fish interact with the world? So we created for the fish in the lab a mini home theater. So our fish is now sitting in this dish, and we are projecting a movie to the fish. So while the fish is watching the movie we are projecting, we are recording the activity from the brain. And a lot of people ask if I show Finding Nemo, so I'll spare the question and I'll say no, he's not watching Finding Nemo. Okay, so I want to show you here, this is the visual brain of the fish, and that's the dish, okay? And you will see what the fish is watching and what happens in the brain. Okay, we see this bar, and again we see a great activity running over the brain, and then we are trying to stimulate the brain with small spots mimicking this food. And what you can see is these large groups of neurons. These are the populations that are active due to this visual stimuli. Okay, so basically, for every stimulus that we present, there's a group of neurons that is active, and the question is, how is this group of neurons coding the information about this stimulus? So I want to go back to this feeding movie just because what we were trying, as you can see, this feeding behavior should be very, very accurate. So the fish has to know exactly where the stimulus is, because otherwise it won't catch it, right? So this is really essential for the survival of the animal to know very precisely where the stimulus is. And this is the behavior we want to study. And so in the lab, what we do is we present the fish with 3 very small spots mimicking this paramecia, okay? And while the fish is watching these spots on the screen, we are recording the activity. Now, if I show you a trace from a single neuron, this is now every dashed line here represents the onset of a stimulus, the different stimulus, and the color represents the identity of the stimulus. So we had stimulus 1, 2, and 3. And as you can see here, this neuron was responsive to all stimuli. So clearly, if the brain only looks at this particular neuron, it has no idea where in the visual field this spot was presented. However, the fish has to catch this food, right? So how does it do it? So this is where we are recording from the population of neurons then. And so instead of working with a single neuron, we are working with vectors of many neurons active. And using various computational techniques, we are trying to find out what is the mechanism by which the fish or the brain holds the information and decodes the information in order to generate behavior. Now, clearly there's a long way for us until we will be able to crack this neural code. However, we can see fantastic implementations already. One of the implementations of us understanding a little bit about the neural code is a field in neuroscience called neural prosthesis. And in this field, what we do, we are trying to drive an artificial limb or artificial device simply by thinking about moving it. And a great example for that was presented at the kickoff of the World Cup in Brazil, where a paralyzed man was in brain-controlled robotic exoskeleton. And what this guy, he's paralyzed, right? He has a helmet on his head, and this helmet records the signals from his brain. Now, this guy thinks about moving his foot or his leg, just thinks about it, and this information from the brain is then transmitted into this exoskeleton, and he actually kicked the first kick in the World Cup in Brazil. So that's one stream where we can help paralyzed people moving artificial limbs. Another very science fiction-y stream that we can start seeing some works around that is connecting brains together. We can assume, let's assume a very complex problem, okay? And I can say, well, I'll solve this part, and you solve this part, and she will solve this part. But if we combine all of our minds together, we will be able to solve it much faster. And we already see some implementations for that as well. And lastly, but not least, another byproduct of understanding this neural code is to build this brain-inspired computing. And this is where neuroscience really interfaces with AI or artificial intelligence. And I'll lead to Geoff to give the last and final part. Thank you.
Okay, let me just get this plugged in again. Okay, so just in the last few minutes, I'm just going to talk a little bit about this subject of brain-inspired artificial intelligence. Now, perhaps the most prominent example of that, which you might have heard of, is called deep learning. Can I just have a show of hands of how many people have heard of deep learning? Okay, so let me just briefly explain. So the way you process visual images, for instance, is in a series of layers, a hierarchical series of layers. So information comes in the eye here, and it's processed through a series of intermediate stages of the brain. So people have modeled that in so-called artificial neural networks. So this is a layered network consisting of a layer of input neurons and some layers in the middle, and then a layer of output neurons. Now, in the last few decades, people have discovered so-called learning algorithms, mathematical ways in which you can train the connection strengths between these layers in order to model particular problems in the world. And in particular, the way these networks work is that you show them lots of examples of this is an input and this is what this input means. And the network is able to extract through these patterns of connections the underlying structure of that problem. So for instance, if you show the network lots of pictures of dogs and tell the network that's a dog, and you show it lots of pictures of cats and you tell it that's a cat, then you can show it a picture of a dog or a cat it's never seen before, and it can classify that as a dog or a cat. Now that sounds a rather simple problem. It's actually not that simple, because the dividing line between dogs and cats is very, very subtle. It's very hard to write down sort of logical rules which will always distinguish a dog from a cat. But the way these networks work is that you don't try to tell the network what to do. You tell it how to learn, and it figures out what to do for itself. So deep learning is one very prominent example. Another example is called reinforcement learning. And so in deep learning, you're just presenting stimuli and getting responses. In reinforcement learning, this is a bit more like the problem that you have to solve in the real world, which is that you have to take a series of actions which you hope will maximize your reward. Now, this kind of problem has been studied for many decades by animal learning theorists. For instance, you put a rat in a maze and you ask, how does it learn where to get different rewards? Now, this may sound like a very simple problem, but humans often face problems like this as well. So what people have done is found that through the combination of mathematicians and animal learning theorists, they've come up with a set of mathematical principles which can both explain how rats learn in mazes and also how you can, mathematical rules which you can use in computer programs to maximize their ability to obtain rewards. And here's just one example of sort of this mathematical formalism. It's called reinforcement learning, and this is a particular example called Q-learning here. And what you're trying to do is you're trying to maximize over all possible policies. So a policy is a set of which action you take in each state of the world, your expectation of the reward you obtain now at time t, and the reward you're going to obtain at all future times, t plus 1, t plus 2, and so on, all the way to the future, discounted by this factor gamma, which is slightly less than 1. So essentially, people have figured out how to efficiently solve these kind of equations so that you can learn, through experience in the world, you can learn to take actions that maximize rewards. And this was inspired, by, as I say, originally by experiments on animals and trying to understand quantitatively what's going on there. So perhaps the most prominent example of AI exploiting this is the work of a company called DeepMind in London. So DeepMind is a startup company that was founded by a computational neuroscientist, and then within a few years it was acquired by Google for about $500 million. And what they've done is they have tried to develop a general purpose artificial intelligence learning algorithm which combines deep learning and reinforcement learning. Now, their first major success was 2 years ago, when they showed how their algorithm could learn to play video games. So they took this set of classic Atari video games from the 1970s, and they took this deep neural network. So the input is the screen, and the output is a possible action. And they showed how this network could learn to play a whole variety of games. So the important thing here wasn't specialized just for one game. It could learn a variety of games. It required a new network for each game, but it didn't require a change in the algorithm. So 1 year ago, they stunned the world of Go by producing a program that beat the world Go champion, Lee Sedol. So the way this program worked is that first they trained it on lots of human moves, and then they let it play against itself to improve itself beyond human performance using this type of deep Q learning. And what I wanted to do is show you a movie. Hopefully we'll have some sound here. So these are the commentators during one of the games.
Yeah, the Google team was talking about is this kind of evaluation value.
That's a very surprising move.
I thought it was a mistake.
So this is a move that the computer made, which had never been seen before at this level of play in humans, and it seemed like a very, very odd move. Now, the computer went on to win that game and resoundingly beat the world champion in a series of games. So already, that version of the program had moved beyond human expertise in Go. Now, the reason Go is interesting is it's actually a very, very difficult game for computers to play. So computers first started beating the best humans at chess about 20 years ago, but it's only last year that they started beating humans at Go. And people thought, most people working in artificial intelligence thought this was going to take a lot longer before computers were able to beat humans in Go. Now, earlier this year, in fact, just a couple of months ago, DeepMind released a new program called AlphaGo 0. Now, what the 0 refers to is that there was no experience with human play in this program. So it completely taught itself how to play Go. I mean, it was given the rules of the game, but then it just played against itself millions and millions of times. And then it actually exceeded human performance. So in the interest of time, I'll skip over this. Now, just a few days ago, they introduced Alpha Zero Chess. So Alpha Zero is their name for their general purpose deep reinforcement learning algorithm. And they found that within 4 hours of self-play, this program was already able to beat the strongest chess computer program. Now, we don't worry about humans anymore in this domain. Humans lost out 20 years ago. So the challenge now is to beat the best sort of conventional chess program, which is called Stockfish. And what they found is that they were able to do that just through 4 hours of self-play. So during this time, it's played hundreds of thousands of games against itself, which is a lot more experience than a human has playing chess. But they were able to show that this algorithm could very effectively learn from that experience. So here is the performance of Alpha Zero compared to Stockfish. So ELO is a measure of the strength of a chess player. And you can see that, so this corresponds to about 8 hours here. Within 4 hours, it already beaten, already was more highly rated than Stockfish. Now, it's interesting because this program rediscovered every standard chess opening that humans have developed over centuries. For instance, this is the Queen's Gambit here. And this shows the frequency with which the program elected to play that opening as a function of its training time in hours. And interestingly, the program started playing the Queen's Gambit more and more often. So this is in its games of self-play. And so in other words, the more it plays, the more it decides it likes the Queen's Gambit. There are other openings where it tries them for a bit and decides it doesn't like them so much. And so humans certainly have a lot to learn about how to play chess from this algorithm. This algorithm also beat, it's actually a more refined version of the program that played Go, and so they also applied the same algorithm to Go, and AlphaGo 0 beat the previous, Alpha 0 beat the previous AlphaGo 0, which beat the previous AlphaGo Lee, which is the one that beat the world champion, and so on. It's just a continuing arms race as their algorithms improve. These programs get better and better. So what's the key point here? That these are general-purpose learning this is a general-purpose learning algorithm, and so there is no additional domain knowledge. So you can apply it to chess, you can apply it to Go, except the rules of the game. Now, this is the key statement here, except the rules of the game. So this is, I think, the big challenge for artificial intelligence. And of course, when you're born, nobody tells you the rules of the game, right? You have to figure them out. So that's one issue, that for all these problems like interpreting visual images, you have to figure it out for yourself. Secondly, even when it comes to learning to play chess, you can learn to play chess based on explicit instruction, and you can become quite good on a tiny number of games compared to the hundreds of thousands or millions of games that these algorithms have played. So these algorithms are very effective in restricted domains where the rules are very clearly defined, and it gets the chance to have an awful lot of experience. So in domains like that, you're going to lose to artificial intelligence, but that's not what most of the world is like. Now, it's interesting because artificial intelligence over the past 70 years, since the 60 years since the birth of artificial intelligence in the 50s, has it gone through a number of cycles. So there was a great deal of enthusiasm and excitement to start with, but then people realized that the problems were actually much harder than they originally realized, and this led to what's called the first AI winter in the 1970s, where funding dried up, people stopped believing in artificial intelligence. Then there was a resurgence of interest in the late 1980s and early 1990s through expert systems, rule-based systems. But then people became disillusioned with them, and we got the second AI winter. So this history is reviewed in an article I wrote for Cosmos magazine, which is coming out online tomorrow. And you can see right now we're in a frenzy of excitement. So you can't open the newspaper without reading about the latest triumphs of artificial intelligence. And those triumphs are real, and we have made genuine progress in the past few years. However, for the reasons I just mentioned, there's still a long way to go in terms of developing a general artificial intelligence. And learning from history, we can see it's almost inevitable that this is going to go down to some extent at some point in the next few years. So how are we going to get over that next dip? Well, myself and many people, including the head of DeepMind, Demis Hassabis, believe that the key to the next set of advances in artificial intelligence is going to be a better understanding of how our actual brains work, because they solve so many difficult problems that we still can't solve with artificial intelligence. And here's a recent article they wrote where they argue that a better understanding of biological brains could play a vital role in building intelligent machines. So that's a problem for doing academic research in computational neuroscience, because all my students get hired by DeepMind rather than staying in the lab to continue their research, because companies like DeepMind are hiring vast amounts of computational neuroscientists because they believe that those are the people who are going to generate the next breakthroughs in our understanding how to build algorithms to make machines smarter. So I'll just conclude by saying that I hope I've managed to convince you that there are very close links between neuroscience, artificial intelligence, and mathematics. And through a combination of those, we'll both develop a better understanding of how the brain works, and we'll be able to build smarter computing devices. So thank you very much for your attention.
Thank you, Geoff. Thank you, Lilach. We'll probably invite you back up onto the stage in a moment. Feel free to grab a drink while you're waiting. We're going to take questions now from the audience or on Twitter. So if you have questions about AI or about the brain or about the impending loss of all our jobs, feel free to write them down, and Leonie and Dom will come around and collect those down the aisle. Just a couple of minutes of housekeeping. We'll, of course, be back next year. Haven't confirmed the start date yet, but probably February or March. So sign up to the website or the mailing list. We're on Facebook, and of course keep watching Twitter, and all of that will be revealed soon. So if you'd like to join back on stage, we've got a question already from Twitter here. So the question is from Ms. Min, and might be for Lilach, but either. The question is a zebrafish decoding spatial info exhibiting the same degree of complexity as a human?
Oh, that's a good question. So I think, in essence, when you look at the circuit, the basic rules and the fundamental computations, they should be very similar, yes. It's clear that clearly, us as humans are dealing with more complex visual scene, but the very basic information processing should be very similar.
Can I just add to that? That the way you make progress in science is by trying to study simplified systems that you can develop a good understanding of how all the parts of that system work, and then you can hope to generalize that to more complex systems. So that's what we're trying to do.
Great. So also on zebrafish, Brian asks, is there any evidence that, based on the zebrafish research, is there any evidence that someone who was born blind has their neurons wired differently from sighted people?
Well, so our work in zebrafish is certainly shows that, at least in zebrafish, that the wiring is different if you don't have normal visual experience. Now, there's been a lot of experiments with animals, with mammals, which have addressed the same question. And certainly, it's very likely that there's a rewiring that occurs in this. If you've lost some sensory input, there's a rewiring that occurs, at least if you lose that sensory input earlier than life. And one of the interesting things about the way the nervous system develops is it's a very sort of Darwinian system, and there's a great deal of competition going on in there. So the fibres, the inputs from the eye are competing with inputs from the ears and so on, fighting for space in the brain. And if you lose input from one sense, then the parts of the brain that would normally have responded to that sense can sometimes be taken over by inputs from other senses. So that's a form of neuroplasticity. So the brain doesn't like wasting its hardware.
Great. Now, I'm not sure if this is in your wheelhouse or not, but Howard asks, what is pain? Other than doing a PhD.
That's a great answer, yeah. Well, pain is essentially, it's just neural activity. It's the firing of particular kinds of nerve cells. This speaks to this sort of almost philosophical question that we really don't understand yet, which is how this neural activity leads to conscious experience. Why is pain painful? Why is a certain wavelength of light look red? We really don't understand that what it is in our brains that gives meaning to this neural activity, a meaning that makes sense to us. And so, I mean, there's a lot of research on it, but we don't have good answers yet.
Right. Okay, so time for the big questions. Stephen Hawking has predicted that AI could mark the end of human civilization. The big question is, how would AI develop morals and ethics?
Well, yeah, that's a good question, and I think we should do it before AI does it. Yes, I think the way to attack it is, obviously there's lots of benefits of using AIs for us as humans, but I can see where things can go to the dark side, and the thing is that we really have to define the borders. And we have to really run with our ethical and morals stuff before AI does it. Want to add?
A lot of researchers in AI are very actively involved in these issues, and a lot of people recently have signed a letter about arguing that there should be a ban on any kind of autonomous weapon systems, i.e. weapon systems that make decisions to kill people without any human intervention, and I certainly very strongly support those efforts.
Right. We have probably time for two more questions before we wrap up. One, which is, do individual neurons work like simple logic gates, like in a computer?
So it's interesting because at one level, that's a question of what is an appropriate level of abstraction in modeling a neuron. So you can model neurons as simple logic gates, either just sort of ones and zeros. However, neurons are actually in some ways much more complicated than that. For instance, they have a complicated three-dimensional structure. And if you look at that level of detail, then they're actually a lot more complicated than individual logic gates. So the answer is, it depends what kind of question you're trying to answer. So sometimes it's useful to think of them as simple logic gates. Sometimes that's not enough for a question you're trying to answer about how the nervous system works.
Right. And the last question is, where can I get the cool sensing kit?
Right, so I meant to say that. So this actually comes from a company called Backyard Brains, which is based in the US. So if you just Google Backyard Brains, you'll find that information. And they're a bunch of very committed neuroscientists who are very excited about the idea of making technology which is normally only available in fancy neuroscience labs available to everyone. So this costs like $200 or so. And it's very easy to use. You don't have to be a neuroscientist to use it, and they have a whole bunch of products.
It's not exactly brain surgery, you're saying.
It's not exactly brain surgery, no. I mean, I have no connection with them, but certainly I think they're doing fantastic work.
Marvellous. All right, we'll wrap up there. As I said, we will be back next year with BrisScience, though I should say, in the meantime, if you'd like to see more of me at least, and you might have seen our talk with Ben Milbourne early in the year, the Food Lab, which is filmed a lot of it out at UQ, is now on air, weeknight 6 p.m. on the Food Network on SBS. So follow up, catch our BrisScience talk from earlier in the year online, and then follow up with Food Lab. Otherwise, could you please join me in thanking our speakers tonight, Geoff and Lilach? Really appreciate it.
Thank you very much.
Please join us outside for some food and drink.